Hello, my name is Robin Deleon, and I'm the Executive Director of Connecting Families UCD Foundation. This video you're about to watch has been produced by Connecting Families UCD Foundation. This video is hopefully one of many which will provide education and information about upcoming therapies for the UCD community. A company named iECURE is close to launching this study involving gene editing for OTC infants birth to 8 months old. At our invitation, iECURE has gracefully agreed to present their current information with us, and we in turn are producing it for you. We would like to thank iECURE for agreeing to be with us, and if you have any questions, you can reach out either to myself at robin@ucdfamily.org or use one of the links provided in the video. Now, I would like to introduce our speakers, Dr. James Wilson, Dr. George Diaz, and Brad Dickerson. Dr. James Wilson serves as a scientific advisor to iECURE. He is the director of the gene therapy program, Rose H. Weiss, and professor and director at the Orphan Disease Center, professor of medicine and pediatrics in the Department of Medicine at the University of Pennsylvania. Dr. Wilson has led the effort to develop the field of gene therapy. He began his work in gene therapy during his graduate studies at the University of Michigan over 30 years ago. And Dr. George Diaz, he serves as VP Therapeutic Area for UCDs at iECURE. Dr. Diaz has more than 25 years of experience in genetics and pediatrics. Dr. Diaz has collaborated extensively with industry partners to bring novel therapeutics to patients with UCDs and other rare genetic disorders. Brad Dickerson serves as VP of Product Management and Patient Advocacy at iECURE. Mr. Dickerson has over 20 years' experience in developing early-stage biotech companies and collaborating with advocacy communities in the execution of clinical programs in the rare disease space. A special thank you to all who are joining us today. And with that being said, let's get started.
You guys can hear me?
Yes.
Yep.
Yeah, all right. I'm going to share screen here. Just give me a second.
Okay.
Let's see. Can you see this?
Yes.
Yes.
Fantastic. Great. Well, listen, it's really exciting to be able to give you guys an update on what we're doing for patients with liver metabolic diseases and in particular OTC deficiency. So I'm going to walk you through a little bit of a science lecture just so that we can understand some basic terminology and some concepts that we're going to discuss with you today. So I appreciate the background and the introduction. And indeed, somewhere around the mid-'80s, I moved with my wife to Boston to complete my clinical training. But importantly, we had the idea that we wanted to develop generic treatments for patients with genetic diseases, and we thought about gene therapy. So back then, the idea of gene therapy was to introduce a normal version of a gene in cells of patients that had mutations in the endogenous gene. And really the conceptual leap in discovery, which occurred while I was at MIT, was the notion of using a virus as a delivery vehicle for a normal gene. And so what I've depicted here is a picture of a virus. Now, we've just been through an unfortunate experience with viruses, that is the coronavirus that caused COVID. But a part of the trick of our trade was to identify the right virus that we could engineer so that when administered into an individual with a genetic disease, that it didn't cause a viral illness. And the way we did that is we eliminated its genes that was responsible for causing disease, and in place, we introduced a therapeutic gene. So this was really, it had a therapeutic payload and it was a delivery vehicle, which we call a vector. So in this setting, this vector, which was based on an engineered virus, would seek out the right cells. In the case of the program we're talking about, hepatocytes, cells of the liver, would deliver the genetic payload into the cell that would migrate to the nucleus of the cell, where it would then take up residence for a while, and we want to talk about durability and what it would mean for the iECURE program. And then once it did, it would undergo two important steps. It would convert the DNA to RNA and then the RNA to protein. And this protein is really the functional entity of the viral vector, in our case, the OTC protein. And this would be an hepatocyte. So this journey started in the mid-'80s, and over many years of work, we spent a lot of time trying to identify what the best delivery vehicle was to deliver genes safely to organs such as the liver. And I will say that in the mid-'80s, that it was my idea to consider targeting genes to the liver for metabolic diseases. And that has been my goal ever since. And it seems like almost 40 years that it took us to get here, but all I can say is I'm glad that we're finally here. But the delivery vehicle, this vector based on a recombinant virus, let me just see here. We discovered around 2000. And this is a picture of a virus. You can't really see it because it's small, but this is a rendition of what a viral vector would look like. And the ones that we landed on around the turn of the century is something called AAV. So AAV is the acronym for the viral vector that we're going to use in the iECURE program. And the reason that we gravitated to this viral vector is that it seemed very efficient at targeting genes to cells, and it seemed relatively safe at the doses that we would deliver this. So with the idea of viral vectors available to us as the tools of our trade, I just want to talk through the work that we did to develop gene therapy for OTC deficiency in the context of another program, and why we felt it was necessary to transition to gene editing. So just as a foundational slide, and rather simplistic but important, that on the left we have a normal cell, a liver cell. On the right, we would have a cell from a patient with OTC deficiency. So on the normal side, we have a normal gene, and this is in the chromosome of the cell, embedded within the cell. That gene produces RNA, which then produces the OTC protein, and that protein is responsible for converting ammonia to urea. And if there's too much ammonia, that it could cause toxicity to the brain or ammonemic coma. So in patients, males with OTC deficiency, there is a mutation in the gene that encodes the OTC mRNA and protein. And as a result of that, there isn't any mRNA and there isn't any protein. And if there's no protein, then the ammonia can build. So again, a mutation in the OTC gene, as a result of that, no mRNA, no protein, and ammonia builds up. So we developed gene therapy for OTC deficiency. And I'm going to describe how that works. So again, we start out with a liver cell that has a mutation in the OTC gene. And this is embedded in the chromosome. It's part of the cell. And so what we want to do is we want to introduce a new version of that gene into the cell to replace the fact that this isn't working. So we'll take a normal version of the gene and we inject it into the patient. And it didn't do what we wanted to do. Did you see that? It gravitated to the cell, but it bounced back because you need a special delivery vehicle to penetrate the membrane of the cell to deliver this normal gene. And that's our viral vector, or AAV. So here's our AAV, delivering the normal version of the gene that makes the RNA, makes the protein, and as a result, there's no buildup of ammonia. That's gene therapy. So let me be clear, there's a mutation in the gene doesn't work. We had a normal version of the gene, and this normal version of the gene is in the nucleus, but not in the chromosome. And as long as that cell stays put, it doesn't go away and doesn't divide, this could lead to a genetic correction or cure of OTC deficiency. So we developed this approach for OTC deficiency. And our plan and our thought was that this would be useful for both adults that have OTC deficiency and newborns. But what we learned, and I want you to please remember that the defective gene is embedded in a chromosome. The new version of the gene is outside the chromosome. That this is a summary of what happens in newborns, that we have a defective gene, we put a normal version of the gene, corrects the cell, but the liver of a newborn divides as the baby grows, right? And the cells divide. And when the cells divide, each daughter cell retains this abnormal chromosome, but only one out of the two cells retains the normal version of the gene. Which means that as the baby's liver grows, we lose the genetic graft because it didn't divide with the dividing cells. So this approach that we developed for gene therapy in OTC deficiency was limited to patients that were adults. You may ask why, and the reason is the adult liver doesn't grow or replicate so that the genetic graft is stable. But in newborns, while it may work acutely, it just didn't work stably. So that product actually we developed and partnered, and I created a company called Dimension Therapeutics that was acquired by Ultragenics, and that AAV gene therapy for adults with OTC deficiency is in late stage clinical development, and I suspect will be approved in a couple of years. But how about newborns? We needed to go to a new approach called genome editing. The iECURE approach is a proprietary and I think very powerful editing technology, and I'm going to illustrate the idea of editing using a similar technology called CRISPR, but it's basically a similar idea. So let's start out with a baby with OTC deficiency. There's a mutation in the gene. As a result of that, the ammonia builds up, the baby is susceptible to hyperammonemia. So what editing does, and this is an example of using CRISPR, is using a viral vector, we deliver a payload, which is called Cas9. And what it does is rather than add another gene, it introduces into the chromosome a corrected version of that gene or a normal version of that gene. So you actually correct the defect right in the chromosome that then leads to RNA, protein, et cetera. So what that means in newborns is this is a cell, a liver cell in newborns, in which there's a mutation in the gene. And rather than adding a separate gene, we essentially correct the mutation in that gene. So now when the baby's liver divides, every cell has a normal version of that gene. That's why we need editing for newborns where gene therapy falls short, because the cells that take the vector up dilute out the vector genome and you lose the effect, where with gene editing, you actually can modulate the chromosomal DNA so that you have a normal version of that gene. So I'm going to stop sharing there and just so when we discovered this approach of editing for OTC deficiency, we were pretty excited because we really thought that we could make a big difference in patients born with neonatal onset severe metabolic liver disease And we needed to raise money to be able to progress this work, although we did a lot of it with philanthropic donations. And then we're able to create a company called iECURE, whose sole purpose is to develop genome editing for liver metabolic diseases, starting with OTC deficiency, and then moving to citrullinemia and other urea cycle disorders, and then other liver metabolic diseases. So I'm absolutely delighted with the progress that we've made. And I have a chance through the Orphan Disease Center to spend time with a number of rare disease groups of patients and families. And this moment in my career, 40-year career, is the most exciting time in those 40 years. The first grant that I got when I started my faculty job over 35 years ago was a grant from the National Institutes of Health to develop a treatment for neonatal onset OTC deficiency. That was 35 years ago. And we finally, after all of that time, landed on an approach with a technology that I think is really going to make a difference. And we did a lot of work, a substantial amount of work in primates and monkeys, including newborn monkeys, to evaluate the safety of this strategy and also the likelihood it would help. Now, these monkeys don't have OTC deficiency, but they can be used as a model. And after those data, we've assembled those data, the team decided it's time to go forward. The iECURE team, including George and others, was assembled, and we are moving as fast as we can to get this through approval from the health authorities, and then to move forward. And I will, and I'll pass it over to George, and then others from the iECURE team to give you an update on where all that is. And I'll stay on the phone, but mute myself. So pass it over to you, George.
Thanks, Jim. Thanks so much, Robin, for the invitation. Great to see you here. There they are. Okay. So I was just going to review a couple of basics to put into context following Jim's presentation. So this is just a little cartoon of the liver. We can go on to the next slide. And we have been talking a lot about genes and pathways. And so I thought I'd kind of be very basic here and kind of start with, the basic problem here, which is a problem of nitrogen metabolism. You don't usually hear about urea cycle disorders as a nitrogen problem, but nitrogen is kind of the most common component of the atmosphere. Next slide. And we actually can't take nitrogen in from the air, but there are other living organisms that do. So plants do, there are bacterial organisms that do, and eventually we get this nitrogen into our bodies. Next slide. And so, for instance, fish are some of the organisms that will utilize nitrogen in the form of amino acids, and we will utilize fish as food. So we will take in that protein. So when you're converting protein, breaking it down into the amino acids, and then using those for your own purposes, for growth and so on, you're making ammonia as part of that process. The next slide. So if you're a fish, you actually don't have a problem with ammonia disposal because you just secrete it out into the water. So fish secrete ammonia out through their gills. They actually don't need to have a urea cycle in their liver as we do. Next slide. But if you have your fish meal, you will start to produce ammonia from the protein breakdown. And so you do need to have a system in place to get rid of the ammonia that builds up. And so this is just a cartoon showing you all of the steps, kind of starting from ammonia here on the left of the circle, and proceeding in a stepwise process where ammonia, which is a highly toxic molecule, is transformed into something that is safer for our bodies to have on board, urea, and urea will travel out to the kidneys and into the urine, and it is excreted. So this is a detoxification cycle. And if you have a genetic change in any of the enzymes that are responsible for the cycle, you will have a problem with ammonia buildup. So the excess of ammonia buildup is really what's the problem. Ammonia is, we have ammonia in our bodies as a normal byproduct of protein metabolism. It's only when this urea cycle is not working that we have an issue with hyperammonemia or high levels of ammonia. And this is clearly most important with regards to the toxicity to the nervous system and to the brain. And so that's the most important issue for us. Next slide. So neonatal onset OTC, again, Dr. Wilson developed very nicely the rationale for why we're focusing on neonatal onset OTC with gene editing. Next slide. So we have a variety of different forms of OTC. As people in your organization know, there are the people that are born and have presentation very early in life. Those are going to be mostly boys, and they have very, very dramatic presentations with very high levels of elevations of ammonia, and they require aggressive medical management. And they have requirements frequently for liver transplantation. So we'll discuss them in a little more detail. But you also have these other buckets where you've got late onset. So patients who are symptomatic, kind of after the first month of life, that can come anywhere from the first months of life to, adolescence, childhood. And the majority of people with this condition are females that we see in kind of the databases. And so these are females who have one X chromosome that has a functional OTC gene, one that is inactive. But here, when you're looking at these patients that are really getting symptoms in childhood, it means that most of the function is not working. So they've inactivated the working copy in their livers if they're females, or they have a mutation that's milder if they're males. And then you've got this third bucket in blue here, which is late onset and asymptomatic. So people who don't know they have OTC until they have some catastrophic event. They may have a surgery, they may have exposure to medications like corticosteroids that unmask the underlying OTC. They've gone most of their lives with no symptoms at all, no medications, no diet, and they've just got this event which can be catastrophic. So we're really focusing just on this first bucket, right? The neonatal onset. Next slide. So I'm just making a point there. Okay, next slide. So again, we've kind of gone through this. I'm not going to focus too much. Again, just to point out, OTC gene is on the X chromosome, one of the sex chromosomes. So girls have two X's, boys have one X and one Y. Next slide. And so the inheritance here is going to be the following. If the OTC change hasn't happened for the first time in that individual that's presenting, if it's an inherited form of OTC, this is the usual mechanism where a mother has got two X chromosomes, one of them harbors an OTC change. She may or may not be symptomatic, right? It doesn't matter with regards to how this is passed on to her children. So in this scenario here, you see a father has a normal X, the OTC is functional. So this couple has risk of having, if they have a daughter, 50% chance of a daughter inheriting that OTC mutation. You can see that mom can pass along either the X that has the OTC change or the X that does not. So for boys, she has a 50% chance of, again, passing along that same, OTC bearing or what we call the wild type or the regular standard gene here. So the father will pass along the Y, doesn't have any input into OTC. It's really driven entirely from the mother, passing along the copy of the X that has a functional OTC gene or the copy of the X that doesn't. Next slide. For males, if you have a change in the OTC gene, as this cartoon shows here, dad with OTC X, all of his daughters will have to have inherited the X from him that has the non-functional or malfunctioning OTC gene, but none of his sons will, because he's passing on only Y to chromosome to sons. So a male with OTC cannot pass along OTC to boys, but he will always pass it along to girls. So this is kind of the general scheme of how X-linked inheritance works. Next slide. And with regard to diagnosis, well, how do you make the diagnosis? Generally, the first test, the most important test, is doing a blood test for ammonia elevations. And this can be really tricky because when OTC presents, it can be very nonspecific. A child is vomiting, is acting sick. When they're babies don't do that many things that give you a clue as to what's going on. And so if somebody isn't actively thinking, okay, let's check for an elevation in ammonia, this is easily missed. So that's the most important first test. And then we've got some confirmatory testing. There will be some changes in biochemistry, but really the definitive test for us is going to be genetic testing. So we'll be looking for changes in that OTC gene. About 80% of the time, you'll find a change in the gene in the coding sequence. So Dr. Wilson pointed out that there are genes have multiple parts. There are switches that turn genes on. There are pieces in between the protein coding parts. If there are changes in there, typically those changes are not going to be identified as readily, and so we might miss some of these changes. You can also identify through genetic testing carriers, so carrier mothers who come in for testing. And if a woman is pregnant and there's a family history, you can also do prenatal diagnosis. So having a diagnosis in the family is super important, because now you can actually pick up risk in subsequent generations. And so, you can be identified through a carrier test before you're pregnant or by prenatal testing during pregnancy. After birth, there are some states that are starting to screen for OTC deficiency. So there are a number of states and territories that are doing this. It's not a great test at the moment, working on making that better as a community, but again, available only in a limited number of geographies. Next slide. So once we have a diagnosis made, what are the basic therapies? So we've been talking up to this point about really advanced therapies, sophisticated therapies. But before we get there, we have to employ the basic therapies that everybody here who lives with OTC knows about, right? Restricting protein. We're going to want to find the right combination of essential amino acid formulas, natural proteins, and that's going to be very individualized for each patient. We're going to provide some ways to try to get rid of that extra nitrogen in the body. So these are the ammonia scavenging agents, and Bufenol or Ravicti are the ones that are commonly used. Most people, were using Bufenol as an oral agent before Ravicti came along. Ravicti has been used a lot more extensively in recent years. During a crisis, you might need an intravenous form. So you might need Aminol, which is phenylacetate plus sodium benzoate and arginine. And this will allow for stabilization during a crisis. But really, for the most severely affected individuals, liver transplant is kind of the standard these days, because it's difficult to manage patients without having subsequent hyperammonemic crises as they grow. So for the severe cases, liver transplant is going to be a consideration. And also, if you have failure of medical management, you may have somebody who's been doing very well on medical management for some time, and then the medical management starts to fall short, and you're starting to get increasing hyperammonemic events. You might consider transplantation in that situation as well. Next slide. So just with regards to kind of what this looks like in the context of a family presenting, this is, I think, again, for the early onset males, the typical scenario where no diagnosis has been made, the family is unaware that their child has OTC deficiency. At birth, everything looks great, and then very shortly thereafter, sometime in the first 30 days of life, in the really severe cases in the first couple of days of life, you'll see a spike in ammonia levels. The children will get very sick, and you'll need to employ emergency measures to get that ammonia under control. Once it's controlled, then you have to get onto a protein-restricted diet and use the diet and scavenger medications to keep the patient stable. And they will definitely, if in very severe cases, probably be referred for liver transplantation. So it's a matter of just stabilizing the patient, getting them to the next intervention if that's what's necessary. Next slide. So again, with regards to this acute crisis, this is where you're seeing all of the urgency being employed in terms of stopping protein intake, giving intravenous sugar and fat to provide a source of energy so you don't get tissue breakdown, which makes the ammonia levels worse, transferring it to a tertiary care medical center for the family if local expertise isn't available there to manage a urea cycle disorder. Scavenger therapy, as we discussed, IV until ammonia control is established, and then you can go on to oral medications. And scavenger medication may not be enough. In the severe cases, really severe crisis, you may need to initiate hemodialysis or some other form of removal of ammonia. So, this, again, very familiar to families who've been in this boat where they see a child born with severe disease or just a crisis due to other reasons. You want to resupply protein after a couple of days to make sure that the body, again, isn't breaking down its own protein supply, which leads to hyperammonemia. And then, kind of figure out what's the appropriate scavenger dose, what's the appropriate protein tolerance for this patient. Next slide. So in addition to this kind of acute presentation, next slide, we might also see a more controlled scenario in cases where prenatal diagnosis has been made. So increasingly, as women are identified that are at risk for delivering a child with OTC deficiency. If that child is identified prenatally, the moms can be actually treated right before giving birth so that the children have some scavenger medication on board when they're born, and hopefully that results in the avoidance of this hyperammonemic crisis that you typically see at a couple of days of life. So in this cartoon, just kind of a little scenario where the child is born, not fed protein, but is put on scavengers, so there's no hyperammonemic event here. The medicines are titrated, so you can figure out what doses are appropriate, how much protein this child needs, and hopefully keep them stabilized without that initial hyperammonemic insult. And then kind of, again, managing to that next step, liver transplantation if they need it. Next slide. So, with regard to ongoing management, we're talking about stabilizing these patients and then kind of moving on to next steps. As most people here know, the real challenges are when you encounter things like infections. So viral, bacterial infections can really destabilize what has been a good equilibrium, a nice steady state. Ammonia levels are in good control. You run into an infection and suddenly the ammonia levels spike. Changes in protein tolerance, which you see kind of at inflections in growth when a child kind of stops growing so quickly, or they start to require more protein or less protein. Those changes can trigger hyperammonemic crises. If you have excessive protein intake, right? So once a child is able to eat, if they go off to a party and they start to ingest protein that they shouldn't, really hard to control that. Underdosing of scavenger medication. So again, we've got to keep up with the dosing, and if a child outgrows their dose, or if there's a medication shortage, sometimes the pharmacy doesn't deliver and there can be a shortfall, that kind of thing. Any of these things can trigger a crisis. Usually when we have a child that's growing very rapidly early in life, that's when they really tolerate more protein than they will otherwise. And so, we've got this honeymoon period where, we have stability for a bit, and then the child will tend to grow more slowly and protein tolerance is reduced. Simultaneously, the child is now getting weaned off of formula. It's really easy to control a child's diet when they're just bottle fed, but then when they start eating solids, and then when they become independent and starting taking food off the table, this is where things become really super challenging for families. And next slide. This is when we might want to consider, liver transplantation. So liver transplantation, the pros here, it is curative for urine dysfunction. You're basically eliminating that problem from your life. There's no more need for protein restriction. You've eliminated the risk for hyperammonemia. I mean, it's really a surgical miracle. It's incredible what surgeons can do. It does come with a downside, of course. The cons here, it's a very invasive procedure, and there's always the potential for complications during the surgery or after the surgery. There's the need for lifelong immunosuppression, which is, again, an essential tool. It allows us to actually introduce this new liver, but it means that you have a risk of infections and some malignancies, kind of going forward. And so this risk benefit always has to be balanced. Next slide. So this is just a cartoon from a paper that was published a few years ago. And it really discussed in a lot of depth, how do families make this decision for liver transplantation? What are the pros and cons here? And just to point out that there are a large number of variables that families have to consider, as well as the providers who are making this recommendation. So this paper actually spoke to providers, it spoke to families, it got perspectives on how everyone was thinking about this decision, which is a very large decision. So, I'm just going to kind of leave it at that there are many, many variables that go into making this decision, and what's right for one family may not be right for another family. So again, highly individualized choices here. Again, liver transplant, an amazing tool for us, but a very, very complicated decision for families. Next slide. So, again, we kind of have gone through the management. Again, this is something that people here have lived and know better than us. We write the prescriptions, but we're not living it. What we want to discuss now a little bit is kind of where we are with regards to moving the preclinical science that has been developed in Jim Wilson's group into the clinical trial environment. Next slide. And so, just a couple of basic considerations here on what are clinical trials. This is a really important set of research studies which really lay out for us whether a drug or a treatment is safe and effective in humans. So a lot of this, all the studies that are leading up to this are done in different types of model systems, but not in humans. So does this treatment actually work, and is it safe? So the way that we actually get to that answer is in a stepwise process. There are different phases of clinical studies, and each phase helps us answer a new set of questions. Anytime we are talking about clinical trials that are current, you can always find information at this website. All trials are registered at www.clinicaltrials.gov. So you can always take a look and see, what are the inclusion criteria, what is the study trying to accomplish. And again, any decision to participate in a clinical trial always should be made with discussion with your physician to find out if it's really an appropriate thing for a particular patient to do. So none of these clinical trials that are on clinicaltrials.gov get there until they have an approved investigational new drug application or an IND. So the research teams will submit all the preclinical data and the study design to the FDA, and the FDA will review it and give the yes or the no on whether the study can go forward. Next slide. So again, in terms of the phases, the phase 1 studies, these are the very early studies where we're looking just at safety. If there's a new therapy developed, is it actually safe in a human being? Is it tolerated? And so this is not necessarily in people that have a disease, but it can be just in volunteers, just looking at safety. Phase 2 studies, so these are still looking primarily, are looking at safety, but now you're starting to look at dosing and efficacy. What is the appropriate dosing to use in a disease population? And finally, when all of that information is kind of obtained, you go to phase 3, where now you're really focusing on efficacy. So this is a trial where you're going to generate significant amounts of data that are focused on showing that this is actually an effective treatment in the study population. So phase 3 is where we determine whether this thing actually works or not. And then finally, phase 4 is the ongoing studies to monitor for safety, make sure that there are no unanticipated long-term effects. The phase 3 studies are generally shorter. Phase 4 studies kind of continue to accrue this data over time. Next slide. So when we're talking about rare diseases like OTC or the urea cycle disorders, it's hard to do this stepwise escalation. So for small populations, we can sometimes combine. And so frequently we'll do a phase 1-2 combined study where we're trying to get safety data primarily, but also get some efficacy data out of this, so that we can try to accelerate a bit, given the rarity of the disease and the difficulty in enrolling these small studies. Next slide. So with regard to where iECURE is, so we are kind of starting to head to the clinic, as Dr. Wilson pointed out. This is the whole purpose of the company. We're really looking at neonatal onset OTC for the reasons that have been described. We think that the gene editing approach is appropriate here. Gene therapy is appropriate for adult onset, but for the infants, really, genome editing is the way to go. This would be a multi-site study, so multiple countries engaged. Because of the small numbers of patients in any one place, we will really need to start looking broadly. We'll focus on genetically confirmed OTC infants with severe onset. Again, those are generally going to be males, and a small number of individuals to be studied at first, less than 10 infants with a multi-year follow-up. So for gene editing, FDA has put out guidance that we're going to have very long follow-up periods to assess safety. So, we are, as a company, committed to engaging the OTC community and taking your input. It's really important that we hear from the community. And so we're really looking forward to continuing to engage and sharing our studies at appropriate time points once we're engaged in these clinical studies. Next slide.
All right, well, I'll spend a couple minutes here at the end of the presentation, just going over our approach and ideas to engaging with the UCD community. Next slide, Michael. So really, our focus is not only on the individual with the disease, but also the family and support system. As George and Dr. Wilson showcased, this is neonatal onset OTC. So this is any decision to participate in research here is going to impact everyone. And we want to be mindful of that in how we're designing and thinking about our studies, where we're doing our studies, everything that goes into that planning. Our team, we're a relatively new company, but the people who are here are very experienced in developing and launching new therapies for rare disease communities. You know, this is a picture of some of us last year at a charity bike ride down at Penn. A colleague of mine, Matt Hall, who was actually leading our efforts to get this trial up and running. You know, we are committed, as George mentioned, to listening, learning, and collaborating with the urea cycle disease community, including Robin, Connecting Families UCD Foundation, and others. We want to engage, we want to hear, we want to listen. We also want to provide support where we can for various community initiatives to raise awareness on things not only about the research that us and others may be doing, but issues that are important to community around raising diagnosis awareness, newborn screening, these types of things. Next slide. You know, and the foundation for how we engage is really driven by something that the FDA looks to companies like ours and others who are innovating, is to engage in patient-focused drug development. You know, what is this patient-focused drug development? Well, it's really engaging with those patients, families, to learn what's important in how we look and study these new technologies, these new drugs, so that we can incorporate this into the way we study and how we evaluate these new treatments. If there are outcomes that are important to a mother for a newborn with OTC that wouldn't be something that a doctor may be looking for, that's important for us to be aware of as a company and to understand. Why is it important? Well, we're not the ones who have the experience, what it's like to live with this condition. That's those in the meeting and their families. You're the ones that are uniquely positioned to really inform companies like ours on what is important in our drug development and evaluation. And if we do this right as a company, it allows us to incorporate these views into these trials that George has talked about, so that as the technology and treatments move forward, that we're analyzing and looking at things that are going to be meaningful when we get through the trials, that these endpoints and evaluations will make a difference to the families. If you're looking for more information on patient-focused drug development, there is a resource at the FDA to learn more on that. Next slide. And so just kind of in closing, before we do get to our question and answer, you'll begin to see more from iECURE as we approach starting our clinical trial, continue to grow as a company. We plan to expand and build resources where necessary, add those to our website. We want to expect and we continue to engage with the community to learn more and listen more. We're really proud to work with Robin and Michael at Connecting Families, UCD Foundation, and others in the UCDC space, and we'll continue to support and partner where we can on these key community priorities. I would ask you to come meet us. This is again my colleague Matt, who was at the meetup, the family meetup back in November in Salt Lake. We will also be at the upcoming meetings this year. I will be attending in person, so I'd look forward to meeting you at these events. And we just have one more slide before we get to our questions and answers. And really what this is a combination of some high-level resources that covers some of the topics we were talking about earlier, whether it was Dr. Wilson or Dr. Diaz. These are good resources to go and learn more about things such as gene therapy and gene editing, as well as the condition, the urea cycle disorders and OTC deficiency. Our website is listed at the bottom of the slide. And as I stated, there'll be a lot more information coming forward as we progress, as we continue to talk with regulators and move our trial closer to becoming and enrolling subjects.
And also, just to let you know too, we will be holding our annual educational meeting, which will be this fall in Philadelphia, and that's going to be October 6th and 7th, and iECURE will be there. And it would be a great opportunity for you guys to come meet them and answer, have your questions answered and things like that. There'll also be other companies there that are sponsoring us too. But, you know, I'll be posting, you know, through social media, when that registration date comes up for you guys to, if you're interested in attending. It's very informative. We will have physicians there, genetic, dietitians, genetic counselors, and speakers like that. So just to encourage you, just to keep an eye out for that. And so I think we're going to wrap it up. So if you guys have any more questions, just reach out to me or iECURE. They did put their website at the bottom of the first, was it the first one? The first one? Yeah. And thank you all for coming. I appreciate you guys taking the time, and thank you to the iECURE team.
Thank you. Thank you, everyone,
Thank
for
you.
coming. Thank you, Robin and Michael, for hosting.
Thank you.
Have a good night, everyone.
Have a great
You
day.
too. Thank you.
Bye-bye.